What becomes of the electrons generated by glycolysis and the TCA cycle? The cell generates ATP through two methods of phosphorylation: substrate level and oxidative phosphorylation. In substrate level phosphorylation, phosphate is moved from a phosphate-bearing molecule onto ATP. Glycolysis and the TCA cycle generate ATP through substrate level phosphorylation. They also generate the reduced electron carriers NADH and FADH2. The cell needs to oxidize these molecules for two reasons. First, it needs to reuse them to continue glycolysis and the TCA cycle. Second, the electrons they carry can provide energy to the cell. The energy in NADH and FADH2 can be harnessed through a different type of phosphorylation, oxidative phosphorylation. Oxidative phosphorylation involves attaching free phosphate to ADP and occurs in an electron transport chain. Electron transport takes a process with a large difference in reduction potential and breaks it up into several smaller steps. The overall process releases a large amount of energy as the electrons move from NADH or FADH2 to an electron acceptor. The electron transport chain uses a chain of electron acceptors to release this energy in small steps, moving protons across the membrane at each step to build an electrochemical gradient. Let's look more closely. The electron carriers form complexes, and each complex along the chain has a slightly higher redox potential. Note that there are many variations in the exact steps and molecules in the electron transport chain. We will look specifically at the electron transport chain as it occurs in the mitochondrion. NADH passes 2 electrons to complex 1, which then passes them to coenzyme Q. The energy released in this step is used to drive protons across the membrane. NAD+ is released and can return to be used again in glycolysis or the TCA cycle. FADH2 carries lower energy electrons and must pass them to complex 2, which passes them to coenzyme Q to continue down the chain, but does not pump protons across the membrane. FAD is released and can be used again in the TCA cycle. Coenzyme Q passes the electrons from complex 1 or 2 to complex 3. Complex 3 passes the electrons to cytochrome C, and protons are pumped across the membrane. Cytochrome C passes the electrons to complex 4, which passes the electrons to the terminal electron acceptor and pushes more protons across the membrane. The terminal electron acceptor can be any molecule with a higher redox potential, that is, a stronger ability to acquire electrons, than the electron carriers in the chain. In aerobic organisms, the terminal electron acceptor is oxygen, which yields the most energy and thus the most ATP. But many alternative electron acceptors are available to anaerobic microbes, such as nitrate, sulfate, or even an organic electron acceptor such as succinate. As the electrons move down the transport chain, the protons pumped out cause a charge and chemical gradient across the membrane. This creates an electrochemical potential known as the proton motive force. This potential can be used by ATP synthase. As it allows protons back across the membrane, ATP synthase spins and phosphorylates ADP, the last step in oxidative phosphorylation. Note that, because fewer protons are pumped out, FAD drives less ATP synthesis than NADH. Similarly, alternate terminal electron acceptors drive less ATP synthesis than oxygen. Outside of the mitochondrion, the proton motive force can also drive other actions, such as membrane transport or the motion of the bacterial flagella. A similar electron transport chain and proton motive force power photosynthesis. We have been discussing the formation of the proton gradient on the inside and outside of a membrane. Where is this membrane found in cells? In bacterial cells, this process occurs across the cytoplasmic membrane. In eukaryotic cells, this process occurs across the inner membrane of the mitochondrion.